last lecture of this course, that is the introduction to good clinical laboratory practice. That is the GLP. So we have tried to cover a large part of ethics. Most of it is on research ethics and publication ethics. We have also covered about good clinical practice in some way, and now we'll be covering about the good clinical laboratory practice. So I request Dr. Raghvendra to take the talk. He's from the Department of Pathology.
Thank you very much, ma'am. So not just in your routine day clinical practice, but also in research, much of the evidence is generated in the lab, right? So when much of the evidence is generated in the lab, so regulators try to, systematize or they try to put some principles or rules in the clinical trials also with respect to laboratory investigations. So now I understand it's lunchtime. I'm showing Maggie. I'm making a big scene. But I just want to drive in a fact that a few years ago, Maggie was banned. Tons and tons of Maggie was buried. And it was said that it was containing high amounts of lead. And Nestle, one of the biggest companies in the world, defended it by saying the lab which tested for the lead in these noodles was not accredited. Now, why lab needs to be accredited? Because when the lab is accredited, it follows a good laboratory practice guidelines. So what is good laboratory practice? So it is a set of principles which involved in the planning of the test, performance of the test, monitoring of the test, recording of the results, archiving the results, and even the reporting of the results. So entire test journey is followed according to the protocol. So good laboratory practice ensures that it is following certain guidelines so that the results that we get can be applied at large populations. So why there was a need for GLP? Long back in 1970s, many of the pharmaceutical companies in USA started producing results for their drugs. And when the FDA took a closer examination into those evidences submitted by these pharmaceutical companies, they found out that either the lab were inadequate to do those tests, or the instruments were not in a proper condition to do those tests, or the results were manipulated. So in order to control all these things, the US FDA came up with a good laboratory practice in 1970s. So Indian government followed it up in 2008 through ICMR, and the guidelines were revised in 2021. So there is only a small difference between GLP and GCLP. GLP applies to all kind of lab, where GCLP applies to only clinical labs, clinical research labs, which are involved in generating results for the application on patients. So and the entire document is available for freely downloading. It's about 180 page. I tried to summarize in a very few slides so that we can understand the gist of it. So what is the intent of GCLP? The intent of GCLP is to ensure the integrity of the data is maintained. There is no tampering with the data. And if the experiments are repeated elsewhere, we will find the same results. Also, the results generated in different lab should be comparable. So to maintain the uniformity and the integrity, the GCLP was introduced. In India, NABL, that is National Accreditation Board for Testing and Calibration Laboratories, has involved in the accreditation of labs, and it is mainly based on the principles of ISO 15189 in 2012, which has the guidelines for following a set of principles if you are giving a clinical report, lab reports. So basically, it tries to address the various aspects, not just the testing, but also where is being tested, who is doing the testing, what are the risk management protocols that they're having, and how they are storing the data. So like in facility, so it provides a guidelines where tests should be performed. What are the infrastructural needs of that should be there to perform those tests? What kind of equipment should be there? It gives a broad guideline about the equipment. It does not say you have to use the same this particular equipment. It just gives a broad idea about what should be the status of the equipment, and as well as the ethical considerations associated with that particular study. And also it is associated with the people who are doing the testing and their constant training and development. And also what is the process involved in the testing, whether it is pre-examination or during the examination of the test results or while reporting it. And also risk involved to the participant, to the performer, and as well as to the environment. And how the documents are stored, shared, and confidentiality is maintained. So as I told you, in infrastructure, it gives a broad guidelines, and it says it should have a separate designated areas and restricted access to the testing place. There should be proper ventilation, environmental controls, and lighting should be there. And there should be proper disaster management plan. Like recently, many trials might have had been done on corona infection. So if good laboratory practice is not followed, then definitely it will be contagious to all the workers, health workers. So proper infrastructural needs should be there to perform those tests. And the personnel, every lab will have definitely an in charge, like a director or a head of the department, and definitely will have a technical person. What is unique here in these guidelines, they say there should be one dedicated quality manager. What is the role of that quality manager? He acts as an in-between. He assists the director in developing the policies and the quality system management procedures, and he tries to implement it through the technical person. So all three kinds of people are required. If you are doing especially in clinical trial, the lab should have all these three kinds of personnel. And it is not just the person's degree, but also it should be constantly trained. Just because one has a degree in, for example, he can do endoscopy, but he has no experience with, he has done DM gastro and he has no actual exposure apart from the here somewhere else. So he should not be allowed directly to do endoscopic procedures. He should be trained. So just the qualification is not enough. He should be trained and then should be involved in the clinical trial. So during the induction, when the person has newly joined the project or the institute, he should be trained according to the job needs. And also on the job training, whenever there is an improvement or a new facility or new technique has been implemented, he should be trained according to that one also. And periodically, the personnel should be evaluated for their competency. So all these documents needs to be maintained. Similarly, for the equipment, so it provides two kinds of annexures. The equipment should be maintained in a periodical manner. According to annexure 1, it should be properly filled. And also, if there is any emergency or breakdown of the instrument, that should also be recorded in annexure number 2. So, and apart from that, the instrument, all the papers associated, like the installation qualifications, performance qualification, operational qualification of that particular instrument when it was installed should be kept. And the logbook of the users who are involved in testing using that machine should also be kept, so that it gives a traceability, confidentiality of the results, everything can be ensured. And regular preventive maintenance and calibration and verification of the equipment is functioning well should be maintained. So there are many kinds of equipments are involved in laboratory testing. We can classify them into 3 groups. Just a simple centrifuge or evaporator comes under group A. You need to ensure visually that it is doing its job. We don't need to be taking this one for calibration, whether it is giving proper results or not. There is no need of calibration as such, but our observation is enough. But in group B, such as a thermometer or blood pressure monitoring system or even microscopes, so it should be periodically checked for its calibration and whether verify it is properly functioning. And group C involves anything which is quantitative, involving software, so like analyzers. So that should be properly calibrated and routinely maintained, as the annexures I have shown in the previous slide. And it is not just the equipment, because equipment is one-time investment, but the reagents or the consumers that one procures should also be of the good quality. It should be procured from a reputed supplier and should be of a standard quality. You should check for the quality marks on it, whether it is approved for testing or not, like FDA approval is there, CE approval is there. So we have to check for those things. And then whenever we purchase a new reagent, it should be first verified it is giving the desired results. It should be checked with the previous reagent that we were using. And always one should label the reagent or the consumable when we implement it in the usage. So that the half-life of unopened reagent is different. Once it is opened, it is different. So it's always desirable if you mark when you open the reagent. And all these logs should be maintained in a register. So if the regulator tries to audit your lab, so then he can ask for these things. So in the testing, appropriate measures to be taken in the pre-analytical phase. Pre-analytical phase just before doing the testing process. So if you are planning to do a clinical trial on hypolipidemic drug and you have not instructed the patient to go fasting while before testing, at least 9 to 12 hours of fasting, the entire thing goes waste. So all these pre-analytical errors, which can be disastrous sometimes. So patients should be properly educated, and when the sample is received at the lab, they should properly check whether it is labeled, if there is any hemolysis is there or not. So other factors which can influence the results should be examined. And during analytical phase, one has to, before actually testing the patient sample, one has to ensure that the machine is giving optimal results with respect to QC material that has been run. So if only if the desirable performance of the equipment is there, then we have to go ahead with the testing of the patient or the subject in the study. And before reporting, one has to again correlate with the quality control report of that particular day with a clinical history and then previous report, as well as with the corresponding lab parameters. It so happens that the creatinine is very high and the urea is not. Maybe clinical situation may be there, but it does not happen in every cases. So you have to check, is it erroneously high values of creatinine has come or erroneously low values of urea is there. So one has to check with cross-check with the other parameters also. And then it should be reported in a format such as in given in the Annexure 5 in the same document which I was mentioning. So all the relevant details of the patient should be noted down and then given signed off with the authorized signatory only. So this is in a nutshell about how to do testing and everything. However, the guidelines also ensure that there should be safety of the person who are involved in testing and the facility also. So one should have take universal precautions such as there should proper laboratory personal protective equipment should be available, and the safety manual should be available at the place of use, not should be locked up in some chambers of someone is in charge. So safety manual should be available at the place of work, and also the standard operating procedures or protocols to do any particular testing should be available at the place of work. It is not just for showing during the time of audit. It should be actually easily accessible for the people who are doing it. So I'm just giving a classification of these microorganisms into 4 groups, just for bring to you knowledge about what safety one has to take while doing research on those things or testing these things. So broadly, by WHO has classified the infective organism into 4 groups, depending on how infective or dangerous to an individual and a community. So can anyone give an example for risk group 1? Any organism, microorganism falling into group 1? First row, second person. Anybody from microbiology here? Any one pathogen you know of which falls into group 1 or group 2 or any group? Okay, just name an organism and tell to which group it falls into. COVID in group 1, 4. Okay. And what about influenza? Influenza falls into 3. Okay. Ebola, anthrax, they fall into group 4. Influenza falls into group 3. And group 2, like fecally transmitted, fecal-orally transmitted organisms such as Giardia. And the group 1 involves like E. coli. So they may not, just by presence doesn't mean that they're causing disease. It depends on the person's immunity also. So it basically they have been classified into 4 groups, and accordingly there are biosafety guidelines or biosafety requirements into 1, 2, 3, and 4. Okay, so if you see the biosafety level 4 has all the tick marks for all the criteria. So what are those? There should be isolated laboratory. There should be proper way to decontaminate it. The air inflow and outflow of the air should be regulated through filters. There should be entry of the persons should also be regulated through double door. There should be airlock. There should be effluent treatment which is coming out of from the lab. Also, autoclave should be available on site. It is not that you send it to central department for autoclaving, and cabinet biosafety cabinets to work on should be there, and personal safety monitoring capability should be there. So in biosafety level 4, all these criteria should be met. So according to the organism that is being dealt with, the biosafety level requirements varies. So once you address the risk management as well as everything, then remains is the quality. The quality is basically doing the right things and doing those things right. So basically it says, say what you do what you say, and prove it. So quality, if you can say hierarchy, so quality control comes first, quality assurance comes second, and quality management comes third. So, you can say so quality is there in everything. So what is the difference? So when you say quality control, it is for that particular assay, that particular test that is being done on that day. Whereas quality assurance, and you will be doing quality control, you will be doing in the lab. While quality assurance is some third party, unknown material will be sent to you or given to you. You do it, perform it, and give the results. You don't know the results. You will be blinded for it. That third party evaluates those results and he gives the performance of our lab. That is quality assurance. Basically, it's a twofold check. Quality control is just checked by the quality manager in the department. Quality assurance by a third party. And quality management is the entire process of reporting or testing or taking the sample and the following actions after reporting. So the entire thing comes under quality management. So quality control, if you are from, I think very few people are from lab, but in quality control in lab usually maintained through this Levi-Jennings chart. So you will give yourself a room of 2 standard deviation for the performance of any particular test. So it should be within 2 standard deviations or within 3 standard. There are certain rules that is applied on it. So basically one has to understand the quality control samples should be run before running any patient samples. And if the lab is doing testing for 24 hours throughout the day, then it should be tested at least every 8 hours, and 2 level of controls. That is a normal level control, normal range, and one is pathological range. If I'm doing testing for glucose, one should have a control value of roughly around 50 to 60 or 80, and then pathological control of 300, 400. So all this we have to check whether the assay is performing correct at both levels. So quality assurance, I've already told, it's a twofold check, and quality management is the entire process, integrating the entire whatever process that is followed in the lab. So it comes to then after doing all these things, you generate data. So in data needs to be, the integrity has to be maintained. So how we can maintain that one? Either paper-based or electronic data. It should be complete, accurate, consistent, and reliable throughout its life cycle. So all the papers should be labeled, or even the electronic data should be, the restriction version control should be there even in the electronic data. If you go to Google Drive, if you are written any Google document, you can retrieve the previous version. That may not happen with the Word or the standalone applications in the computer. But if anything which is connected to cloud or anything, you can retrieve the previous version. So, and then confidentiality, access to the data should also be controlled. It is not just any person in the lab is going to access the details for the patient and the results, but it should be controlled according to who can access to which level and who can edit the results also. So that should be maintained, and that comes under data security. So data security, not just hardware, that is access security and network security, but also about the applications security. So if someone is involved in pathology, and he should be restricted to only looking at the pathology results, so that she should not be blinded with the other clinical details if the study requires. So the application specific security also should be there, and personal security should also be there. So in a nutshell, the GCLP, that is good clinical laboratory practice, tried to address not just the testing facility or the testing person, the risk associated with it, the data which is generated out of it, and also the infrastructure associated with it. And the intent I already told you, for the reproducibility of the results. So if someone performs the same test, same trial in some other place, should be able to repeat the results. So that is the whole idea, with minimal risk to the participant and to the performer. So entire thing, the 180 page document, I'm trying to tick off most of the things, and hope I think it ticked the right parts of your brain also. Thank you.